Hello and welcome to another Piper Pod. Today we're going to look at the male reproductive system. So moving right into it, the role of the male reproduction system is pretty simple. Okay, it literally only has two jobs to do. Okay, it's responsible for the production of sperms and the production of the male sex hormones, okay, called androgens. And that's it. That's all the male reproductive system has to do. So today we're just going to focus on, obviously, the production of both and how they relate to one another, and then how each one of those particular roles plays a part in the overall reproduction of life. So first of all, let's just have a good look at the gross anatomy of the male reproductive system. So it's a unique system. Much like most reproductive systems, it's quite dormant most of the life. Okay, so when you look at all the other body systems, it essentially starts from birth and it has slight changes throughout your development, but it pretty much just is working throughout. Whereas the reproductive system, in both males and females, is essentially dormant right at the start, and then it peaks around puberty. Okay, so the male reproductive system has internal structures and external structures. Okay, the external obviously being the scrotum, the testes, and the penis, and internal we have our vas deferens, prostates, and seminal vesicles, and obviously they play a role in the production of semen, which we'll go through. Now, the testes themselves, they're responsible for the production of sperm and your androgen hormones. There's two times in life where you have a peak of androgen production. One is during fetal development, where you have that time period as you grow in the uterus, where you have to distinguish whether or not you're going to be male or female based on your genetic code. What will happen, testosterone mostly will be produced, and you'll start to grow male features within the actual uterus. And then obviously as puberty comes along, testosterone kicks in again, and we start to develop from a boy into a man. Okay, so we start to develop those classic masculine type features, okay, where we have larger muscle mass, bigger bones, facial hair, larger larynx, all that sort of good stuff happens because of the influence of testosterone itself. Now, having a closer look at the scrotum, the reason that it all has to sit on the outside of the body is that sperm only survives and only can be produced if it's about 2 to 3 degrees lower than the rest of the body. Okay, so it likes to be around about your 34 to 35 degrees within the scrotum itself. Okay, that's how spermagenesis occurs. So as the temperature changes in the person or in the environment, we have specialized muscles that can retract the scrotum and therefore bringing the testes up closer to the body, or extend the scrotum, therefore bringing it further away. Okay, so this process needs to happen in order for spermagenesis to actually occur. Okay, so whilst it's not a very good survival strategy to have your reproductive organs exposed on the outside, it's a necessary state in order to actually produce sperm. So when we look at a particular testes, so the testes itself is the gonad of the male. Okay, it's about 2 inches long, and we have two of them, and they're surrounded by connective tissue. Okay, but on the inside of this connective tissue, we have a whole bunch of separate compartments. Okay, so 200 to 300 separate compartments within the testes themselves, and each one of those compartments is filled with 3 to 4 seminiferous tubules. Okay, so each one of these seminiferous tubules within these compartments where sperm is produced. Okay, it's also where the specialized cells that produce testosterone are also. But what happens is sperm starts its formation within the seminiferous tubules themselves all around, and then it moves into the actual collection port, okay, the tubules of Retes. Okay, and then from there they travel out to the epididymis where they're stored. proper sperm is stored within the epididymis, and from there it travels up through the vas deferens until it gets to the specialized glands that turn sperm into semen, which we'll go through later on. Okay, so just want to mention that the actual production of sperm starts in the seminiferous tubules. As they develop, they get stored in the epididymis, okay, and then they get to travel out. Now that entire process can take about, what do they say, about 68 to 72 days. Okay, so it's not a quick process. So the storage of sperm is obviously necessary, otherwise you'd never be able to ejaculate, never be able to have children, unless you want to wait 72 days. So we have the storage of this sperm as a backup, and this process is always happening. We're always producing new sperm and then storing it, releasing it, storing it, that sort of thing. Okay, so there's a specialized structure within the testes that we'll go through that help this process obviously stay protected and obviously form. Okay, now the production of sperm is a process known as spermagenesis. So if we were to get one of those seminiferous tubules, okay, and we look at the actual membrane itself, so we look at the actual structure of the seminiferous tubules itself, what we have on the outside is a layer of smooth muscles. Now this allows for peristaltic action to occur. Okay, then just below that we have a bunch of specialized structures. Now realistically, these specialized structures are mostly your serotoli cells. Okay, so your serotoli cells are the entire brown part. The serotoli cells are the ones that help the production of your sperm itself. Okay, so they're responsible for that. They also have a few other specialized roles, okay, but the entire thing is the cell, and where the thing is pointing to on the right is just the nucleus itself. Okay, so within the serotoli cells we have these other specialized cells that are called spermatogonias. Okay, now spermatogonias are essentially the starting point, the recipe, if you will, for the production of sperm. So they start right up the top at the base membrane, and then they wait until they need to be produced. Now, in the influence of testosterone, they get produced and they start to go through the process of spermagenesis. So the first process that these spermatogonias go through is they go through a process called mitosis, where they split into identical cells and become a spare spermatogonia and a spermatogonia that is going to become sperm eventually. Okay, so they have two different types. That way they will replace itself. If they didn't go through the initial first stage of mitosis, you would run out of spermatogonias and that would be it. So the production of sperm, believe it or not, only changes a little bit as we get older. The influence of testosterone changes a lot, but the actual amount and the ability to produce sperm doesn't change throughout life. Okay, well, not very much anyway, okay, because of this initial step of mitosis. Once mitosis has finished, then we have two identical cells. Okay, now type B spermatogonias actually differentiates a tiny little bit as it becomes a primary spermatocyte, develops extra chromatids. Okay, now I'm not going to go too much into chromosome development because we're going to talk about genetics in our next presentation, but at this stage we have obviously our 46 chromosomes, but 4 chromatids itself at this point. All right, from there we have a split into secondary spermatocytes. Okay, now this split here is called meiosis, okay, not mitosis. Now essentially, for simple terms, the difference is mitosis makes up 2 identical copies, whereas meiosis splits the genetic material evenly across 2 cells. Okay, so that's it. So at this point, after this first meiosis, we have 2 daughter cells that both have half the genetic material and 2 chromatids each. That's it. Okay, now from there they actually migrate through an area called the tight junction. Okay, now this is a specialized area within the actual serotoli cell, and this tight junction is actually what protects the sperm from the immune system. Okay, because as you could imagine, now you've gone from a identical identical spermatocyte into secondary spermatocytes. Okay, with half the genetic material, which technically is not the same as the rest of your body. So your immune system will actually see daughter cells as a foreign body and destroy it. So if it wasn't for the tight junction creating the blood-testy barrier, then all of the gametes that were produced would be killed by the immune system. Okay, now this is what happens when you have vasectomies. Okay, after about 3 to 5 years, you'll actually start producing antibodies for your sperm. Okay, because the tight junction in that particular area that's been cut and tied off is not as good, so therefore the immune system can start producing antibodies for it. Okay, just as a bit of a side note. So that's tight junction is very, very, very important. Okay. Now remember, all this is being aided by the serotoli cells itself. Now we'll have look a bit more about the hormonal function in all this in just a little bit, but just so you know, there's structures that are aiding in this whole process. Alright, so after meiosis 1, we have 2 secondary spermatocytes. From there we go through a process of meiosis 2. Now this meiosis 2 is slightly different, because obviously I said meiosis was splitting down the middle. Okay, now obviously meiosis 2, you've still got the same amount of genetic material. You've still got 23 chromosomes within each one of those daughter cells, but now you've only got one chromatid. Because remember, you had 4, then each cell had 2, well now you have 1. Okay, that's why there was that difference at the start. Okay, now meiosis 2 is actually what starts the process of production of spermatids. Okay, so from the 4 daughter cells, they start to develop into sperm themselves. Now the way they do that is they dump all excess cytoplasm and material that they don't need. They form a specialized head, a very cool midsection, and they create a flagellum themselves. Okay, we'll have a little look at the anatomy of the sperm itself, but that's what they do. From there, they get pushed into the lumen of the seminiferous tubules, and they go through the process of moving through the stages towards the epididymis itself. Now this whole process here, just this little bit here, can take around your 50 to 60 days to do. Okay, so whilst we went through it and it seemed pretty quick, it's about 50 to 60 days to get from the initial spermatogonia all the way down to sperm production. Okay, so another cell that we haven't really spoken about is the interstitial cell. Okay, called that because it sits within the base membrane. Okay, its other name is the Leydig cell. Now that is a cell that is responsible for the production of testosterone that aids in this entire process. Okay, when we look at a sperm, this is the end product. This is what's produced from one of those normal cells as it all comes down. So obviously the head has a nucleus that has half the amount of genetic material it needs. Okay, so that's it, half, because obviously when it joins with an egg that has the other half, we create a human. Okay, nice and simple. Right on the top of it, the acrosome, is a specialized penetrating area with digestive enzymes, basically, that allow you to penetrate the egg. Okay, so you know how you say that one sperm wins the race? Well, it's not actually the case. Okay, so hundreds, if not thousands of sperm win the race and weaken the side of the egg wall as it comes through. Weakens, and then eventually one opportune sperm, basically, when the egg's weak enough, comes through and finally gives it the final blow that allows it to go in. So the amount of sperm that's produced during an ejaculation is anywhere between 50 million and 150 million sperm that are released during an ejaculation. Now, the body registers low sperm production, okay, or infertile sperm production as 20 million per mil of sperm that is produced. Okay, so we're talking about enormous numbers of sperm that gets released. From there, we have the mid-piece. Now, the mid-piece is a highly compact mitochondria base. Okay, and this allows for rapid energy production to allow enough energy for the flagellum or the tail to be fueled in order to move so the sperm can spin. Okay, so let's have a look at our hormones. So obviously this whole process is coordinated by hormones, okay, it's a feedback system. So in the presence of low testosterone, or the need for more testosterone, or the need for more sperm, the hypothalamus secretes the gonadotrophic releasing hormone. Okay, that's obviously one of the stimulating hormones that gets released to the anterior pituitary gland, and from the anterior pituitary gland, they release follicle stimulating and luteinizing hormones. Now, I know you guys have heard of these before, but most of the time when we speak about these hormones, we're speaking about the menstrual cycle. So both of them actually play a role in the production of testosterone and the production of sperm. So we actually have a condition or a state known as the hypothalamus pituitary testes axis, which just means that each one of these hormones that are produced, if one of them is out or if something influences them, it actually has a relay effect on the other two as well. So if you produce too much hormone or too much testosterone, and essentially the message is fed back to pituitary to stop, which is fed back to the hypothalamus to stop as well. Okay, so it's a proper axis, and they work together in order to produce the hormones that we need. Okay, but we'll stick with the left side for starters. So we'll look at what the follicle stimulating hormone does. So that stimulates the Sertoli cell to do one of two things. The first one is the production of an androgen binding protein. Okay, now this aids in sperm production. Okay, so if the androgen binding protein wasn't there, as the interstitial cell or the Leydig cells produced testosterone, then just through normal diffusion, the testosterone will be spread between the testes and the blood. Okay, what the androgen binding protein allows is for higher concentrations of testosterone to stay within the testicles themselves. Okay, so the concentration is actually 20 to 50 times the amount of testosterone can be found in testicles compared to blood because of your androgen binding protein. Okay. Now the luteinizing hormone, that stimulates the interstitial or the Leydig cells, okay, and that produces your androgens, okay, the main one being testosterone. Now as the testosterone levels builds up in the blood, it gets a little bit too much, it actually has a negative feedback system, and increased testosterone will actually cause the release of a hormone called inhibin, and that there turns off the anterior pituitary gland, if you will, and no more luteinizing hormone is released, or a reduction in luteinizing hormone is released from there. Okay, so once the sperm is produced and it's gone through the epididymis, from there, during sexual stimulation, the sperm will actually move up the vas deferens. Now, vas deferens goes over the top of the bladder, and then it comes down. Once it meets the seminal vesicles, about 60% of the total semen is produced there. Okay, so sperm only makes up about 5% of the total volume of semen. So 60% of that volume, it comes from the seminal vesicles themselves. Okay, so you've got two seminal vesicles, and they secrete this fluid into it. Now this fluid is a highly alkaline solution, and it has a whole bunch of fructose, energy, and prostaglandin to aid in the sperm survival as it travels into the vaginal passage. Okay, so the vaginal passage is extremely acidic by nature, okay, because it needs to be in order to protect itself. So if sperm was to be released out of there, then without the alkaline substance, then the sperm will die within the acidic environment. Okay, so that's basically it. Now it's also got full of sugar. The mitochondria of each sperm can actually have enough fuel to travel along. Okay, because it is a very long distance in which they have to travel, and it's a very harsh environment as well. As it comes along a little bit further, okay, the prostate is also stimulated, and that secretes your milky 30%. Now this one is slightly acidic, okay, and within this particular fluid as well, it has any bacteria within the actual semen or along the tract to be killed, so they don't end up using the sperm as its fuel source. Okay. Now the last little gland that we have, okay, two of them, the Cowper's gland. These ones here, they can become part of the semen itself, but most of the time, this is what produces the pre-cum. Okay, that's what we always call it. So the Cowper's gland comes out and it secretes this clear solution, and it alkalizes the urethra. It kills any bacteria that's found within the urethra, and it essentially lubricates it as well. So when it comes to the point of ejaculation, the semen itself has a clear, alkaline, smooth runway, if you will. Okay, so that's what the Cowper's glands do. And all of this is obviously controlled by neural stimulation, which we'll have a look at. Okay, so once we get to the point of needing to use sperm, obviously the first thing that happens is the erection is formed. Now an erection comes mostly from sexual stimulation, okay, but as the boys know here, it can also happen for no reason at all. You can actually get an erection without your brain even really knowing about it. Okay, it can happen as a base of baroreceptors within the bladder being too high, vibrations coming through your legs. Doesn't really need too much of a stimulus in order to get an erection, but it's controlled by the parasympathetic pathways. Okay, so this is why when we look at things to do with spinal injuries, why priapism is a thing, because the actual erection itself is caused by the parasympathetic nervous system releasing vasodilatory substances, which causes a whole bunch of blood to come into the penis. Now, as it grows and hardens, the veins will clamp off the back in order to hold the erection for longer. And that's how the whole erection side of the house starts. So from there, with increased sexual stimulation, then the sympathetic nervous system comes into play, and that there actually starts contracting your smooth muscles. Your smooth muscles of your epididymis and your vas deferens, and that causes the sperm to move towards the start of the urethra. It's the sympathetic nervous system that also essentially contracts the larger muscles around the penis itself and causes the smooth contraction of those glands that we just spoke about to release their fluids to produce semen. Now once we get to the base of the urethra, it is a single forceful contraction of the urethra that causes the ejaculation itself. So once you get that massive contraction, ejaculation will occur, okay, and it's designed that way in order for the semen to optimize release to make it all the way to the cervix within the female in order to be picked up as the cervix relaxes. Okay, if it didn't have the forceful release, then obviously there's a risk of it not reaching the end of the cervix. Okay, so that's the male reproductive system. I hope you got something out of it. From here we will move into part 2, if you like, and we'll talk about the female reproductive system. Okay, so until next time, take care.